Okay, happy Monday. So remember, we do have an exam on Wednesday. Today's lecture will not be covered on Wednesday. All right, so it's chapters 10, 14, 15, and 16, which is plenty for Wednesday's exam. There's 70 questions. I'm still giving you longer than a minute for every question. So I still don't think you're going to be crunched for time, even though there's a few more questions than last time. I did just this morning post some practice questions, 10 practice questions. So if you go under the tab on Blackboard that says exam objectives and practice questions. So you'll find those there along with the answers. So take a look at those. All right, and in addition, I've been given a few practice questions as we go along here. This question gets at what's probably the most complicated thing that we've done so far, T-cell activation. So think about the kind of things that I could ask you about that. Like, I'm not going to make you, I can't ask a multiple choice question and ask you to explain it all. All right, so this question, cytotoxic T cells, how are they activated? All right, I'll give you a second to look at that if you haven't had a second to do that. So hopefully you could kind of narrow it down to one of the first two here. Okay. The perfin granzyme pathway here, that's what cytotoxic T cells do to actually kill our cells, our host cells that are infected. This may play a role, but it's not the best answer there. Antibodies are not ever going to play a role in activating cell-mediated immunity. The correct answer here is answer B. So T helper cells basically have to get activated. They differentiate into T helper type 1 cells. They release cytokines that act on cytotoxic T cells with T cell receptors that recognize the same antigen or epitope of antigen there. Okay. And one more to practice. All right. All nucleated cells in the body have the ability to advertise endogenous antigen on the surface of their cells. How do they do that? This is one of those things that you really just have to memorize. There's a number of things you can think through, but this you just kind of have to memorize. They're going to advertise endogenous antigen by MHC class 1. Remember, one hand, MHC class 1, that's what's kind of volunteers what's going on inside of the cell. These answers get increasingly more silly this time. All right, so you have to kind of just remember it's that all the nucleated cells have MHC class 1. It's only the antigen presenting cells that can present with MHC class 2. They're presenting what they phagocytize. Okay, so that's macrophages, dendritic cells, as well as B cells can do that. All right, so let's move on. Hopefully this is a topic that's of interest to you right now, right? So today's the day that your age group gets the go-ahead. I know a lot of you have been vaccinated already, but anybody that's not a healthcare worker or whatever, just anybody that's 16 and up can now be vaccinated. I just want to say, and I'm gonna try not to preach at you today. If you want to get the vaccine, think about which one you're getting, what you're signing up for. Because if you sign up for the Pfizer vaccine, that's going to put you getting the second dose during finals week. All right, so make sure you schedule it so you're getting it maybe at the end of finals week, because plan the day after, you're probably not going to feel so great. All right, so at the end of the semester, if you're too sick to take a final, that starts to present a problem, right? So keep that in mind. I had my second dose of Pfizer on Thursday and I didn't have too much trouble. So I had a little bit of headache, but not too big a deal, but it's different for everybody. If you can possibly get the Johnson & Johnson, you know, one dose, get it over with, whether you're traveling or whatever. It's not going to present a problem to be back here for the second dose. So I've heard that Family Fair is offering Johnson & Johnson, and there is one just a couple miles away out Lake Michigan Drive. So I also, just while I'm thinking of it, I had a student that works at Walgreens Pharmacy, and she said they have all these appointments that nobody is taking advantage of. So, you know, whatever, Walgreens might be an easy place to get in. If you go downtown to DeVos, it's just a big rigmarole, you know, parking and getting in. It's just a huge operation down there. But again, I would encourage you to do that, definitely. So immunizations, there has always been a little bit of hesitancy to towards immunizations, right? So I just find this picture really fascinating. This was put out by the Anti-Vaccination Society in 1824. So it's, you have to look at it kind of closely. I don't think I posted this, but you know, people are taking on bovine characteristics after being vaccinated. And of course, it's referring to the fact that the vaccination that they were doing then was to smallpox. Edward Jenner is given credit for noticing that milkmaids that have had cowpox didn't very often contract smallpox. So he took some scabs from cowpox, which is kind of a mild version of smallpox, we don't know, and inoculated them into a young boy's skin. So in that, he didn't have syringes then, he just took a knife, poked him in the skin, put some of those scabs in there. And then, this is not the way we do clinical trials today, but he intentionally inoculated the boy with smallpox. And he didn't get smallpox. So that started, that was the first vaccination. You know, people had some reason to be afraid, right? We didn't understand how that vaccination worked. So it was a little bit more understandable. But of course, you know, people didn't throw up cows or didn't have cows coming from, you know, other spots of their body. But, you know, again, people are afraid of things that they don't understand. There was some reason to worry. I mean, people could potentially get, you know, some kind of staph infection from being inoculated, you know, with a presumably maybe dirty knife. So there was some complications from that. Smallpox has about a 30% mortality rate. So still, taking your chances with getting inoculated this way would have been good risk management. Okay. Vaccines have gotten safer and safer since then, though. So I want to talk a little bit about, you know, why we vaccinate, why it's important, and the different kinds of vaccines that are in use today. Right, so that term vaccination, we kind of use that synonymously with the term immunization. Technically, to vaccinate means to get a shot, and immunization is what happens inside our body, like our immune cells get activated. Right, so technically it's most correct to say when you vaccinate, you immunize, but again, we kind of use those terms back and forth to mean the same thing. Okay, so it's kind of obvious why we would vaccinate. Obviously, we want to protect people and keep them from getting sick in the first place. Probably the biggest group that we want to protect are children. We just talked about there's certain kind of antigens, T-independent antigens, that young children don't have a good response to T-independent antigens. All right, so these are ones like that Haemophilus influenza I talked about last time, can cause bacterial meningitis in children because their B cells are just not very mature yet. Okay, so adults have a good immune response to that. So children are more susceptible not only to B-independent antigens, but lots of different infections. They're more likely to just have a more serious case. Okay, when you get whooping cough as an adult, it's a serious infection, but it's not likely to kill you. It's usually, you know, young babies that are susceptible to the worst outcomes with whooping cough. So we want to protect children. We know that as we age, our immunity wanes a little bit. So older people are more susceptible to having severe forms of infection. especially things like pneumonia. So we have a vaccine that's recommended for people, I think it's either over the age of 60 or 65, to try to prevent certain kinds of pneumonia. Sorry, we'll talk about that later. Other things like shingles, we have a vaccine that works the one we have now works really good to prevent adults from getting shingles, older people. So that's important. And it's important for those of us that are, somewhere between the extremes of life, because we want to try to get to herd immunity. So this is this term that you've probably heard a lot about if you paid attention in the last year. Herd immunity just means as long as you can get most of the people in a population not susceptible anymore to the infection, then the few people that are susceptible are likely to be protected, because the disease wouldn't likely come in contact with those people that are not protected. All right, so there's lots of arguments right now what it will take to get us to herd immunity for COVID, because some camps will only count people that are vaccinated as being immune, and others, well, the people that are sick should be immune as well. The question is, are they immune to the variants as well? By most estimates, we need to get up around 70 to 80% to have good herd immunity, so that the people that are not able to get a COVID shot because of some health reason or allergic reaction to previous immunizations, so those people will be protected. And of course, we're not vaccinating kids yet, so it's likely to be a while till we can get, whatever, 80% or whatever of the population vaccinated. What percent do we need protected before everyone's protected? It depends on the infection. For measles, we need to have on the higher end almost everybody vaccinated, because that infection is so transmissible. There's a number called the R0 value that if you took an epidemiology course, you would get into that a little bit, but that's kind of a number for our best guess of how many people you would infect, how many people an infected person is likely to infect. So that R0 value for measles, one person that's infected is likely to infect 12 to 18 other people. All right, so, and that's, if everybody was susceptible. So, with COVID, they've tried to do like their best guess of what that infectability rate is somewhere between 1 and 4. All right, so it's hard to take into account, we're on lockdown, what's that number going to be, one person infected, or if some people are vaccinated and some people aren't, somewhere between 1 and 4. So if you get COVID, somewhere between 1 and 4 other people are going to get COVID from you. All right, so that's a good reason to get vaccinated, to protect people around you. All right. All right, and of course, the goal of vaccination, we want to induce B and T memory cells. Okay, so hopefully we get such stimulation that those B cells and T cells make memory cells, and then if we are exposed to COVID, or again, whatever that pathogen is, we don't have to go through the whole activation process. We start making antibodies right away, and the person doesn't even get sick at all. Okay, so we need good adaptive immunity, in other words. All right. So let's look at the types of vaccine that are approved by the FDA to be used right now. Okay, I had to expand my list a little bit this semester. So all the vaccines that we use in this country, up until now, were one of 3 different kinds of vaccines. Like I said, I had to expand my list a little bit for today. First kind of vaccines are attenuated vaccines. All right, so this is the oldest vaccine that we've had. Attenuated means weakened. Okay, so we have Or in other words, this is a live, the pathogen is alive. Or if it's a virus, right? Viruses aren't really alive, but the virus is not going to cause a full-blown infection. It may cause kind of a weaker form of infection, but it's been modified so it can't cause that full-blown disease. All right, so how do we modify it? There's a lot of different ways we do that. Sometimes it's grown in a cell type that's not its normal host. For example, for the Sabin poliovirus, we grow it, normally that grows in human neural cells or nerve cells, and we grow it in monkey kidney cells. Okay, so somehow that alters it a little bit so it's not able to reproduce in our cells anymore. Sometimes you just grow it for many generations of cells. That's the case for the tuberculosis vaccine. Sometimes you grow it at a higher or lower temperature. All those things can, again, cause slight alterations so that it can't cause full-blown disease. And by the way, I talk about a lot of examples here. I'm mostly using those as they're just examples. You don't have to memorize, oh, what kind of virus or what kind of vaccine is the polio virus? You don't need to know that, but I do want you to understand what it means when I say an attenuated vaccine. Okay, that's some kind of weakened strain. These are good vaccines in some respects, not so good in others. All right, so advantages there that you get kind of a more natural adaptive response because that, say, virus is still able to infect cells. We get, you know, a normal kind of MHC presentation of antigens, and it's just a more, again, natural immune response. And so we usually get really good memory from any vaccine that's attenuated. It can, like I said, form a very mild form of the disease. So that can be good if you're trying to vaccinate a very remote population, say, you know, in the jungles of the Amazon or whatever, and you vaccinate most of the villagers. Some of them are off hunting or whatever, and you don't get to them. But, you know, maybe if some of the villagers have kind of a mild form, they can spread that to others that weren't vaccinated. We call that contact immunity. All right, so you could consider that a good thing. Really, we don't want that to happen, at least in the United States, because you could consider that a disadvantage as well, because we don't really want people getting sick, because you don't want to pass that even a mild infection on to a baby or somebody that's immunocompromised. Okay, so we're going to call that a disadvantage as well. Possible to have transmission. All right, so none of the attenuated vaccines that we use, as far as I know, that's not possible. Another thing, because the pathogen is alive, we have to refrigerate it, the vaccine. And that's not so much a problem in this country, but again, if you're trying to go to some remote location in the tropics somewhere and trying to keep it cold, might be a huge disadvantage. One thing, did I mention this? Sorry, I forgot to mention this as an advantage. We typically need to give fewer doses. Sorry to make you flip around there a little bit. But usually, you know, maybe one dose, a booster a number of years later, and somebody with an attenuated vaccine is going to be immune for life. Okay, but the biggest disadvantage to an attenuated vaccine is that there is the potential to have some kind of reversion. And this would be a very, very, very rare event. But so to give you an example, for years and years back, I think it was in the 60s, oh no, late 50s, we had two different polio vaccines that came to be developed about the same time. The Sabin polio vaccine was attenuated. So that was one, it was a good vaccine, good protection against polio. That was the one that we started using in this country. The Salk vaccine, which is a type of vaccine we're going to talk about next, it was a killed version of the virus. That got more popular in other countries. So both vaccines were used. We stuck with the Sabin one. It's kind of nice because it's one less needle prick for babies because you could give it orally. So it was just in a little vial that, you know, babies could kind of suck down. And my daughter, who's 25 now, I can remember she had that, you know, in the mid 90s. Well, in the year 2000, they had a case of it's like 8 to 10 kids that actually got polio from the Sabin polio vaccine in the Dominican Republic and Haiti, I think. All right, so somehow this weakened form kind of, there was some mutation and it reverted back to the wild type. Okay, so reversion back to that wild type, and kids got full-blown polio. All right, so obviously we can't have that happening. So ever since then, we no longer use that attenuated polio Sabin vaccine. We use the Salk vaccine. Okay. All right, so again, just some examples. The MMR vaccine is actually attenuated measles virus, mumps virus, rubella virus, yellow fever. Because of this potential reversion problem, we don't use attenuated vaccines unless there's no other choice. We tried to come out with a measles vaccine that was a killed virus. That was the first one that came out, I think, in the 60s, and it wasn't good enough. It wasn't very effective. We didn't form good memory from it. So again, we don't have anything that works better. This one works really well, and it's very safe. Chickenpox, I think, is another one that's attenuated. Right. Probably the most common type of vaccine in use is some kind of killed vaccine. We also refer to these vaccines as inactivated, again, because viruses, well, are they alive to begin with? I don't know, it makes more sense to call them inactivated. Okay, so some advantages to this kind of vaccine. They're not able to replicate in the host, right? Because if it's a bacterial cell, it's dead. All right, we've done things to treat it, to kill it. I talk about this down here. Hit it with some radiation, hit it with some kind of disinfectant like formalin, heat it, bake it. Okay, those are all ways that we can inactivate these vaccines. So they're dead, they can't replicate in the host. You cannot get the flu from a flu virus, or sorry, vaccine. You can't get the flu virus from a flu vaccine. A disadvantage is that you often need more doses of it to get a good level of protection. So maybe a series of 3 to 4 shots, you know, over a number of years. And because the pathogen is dead, they're not taken into cells normally. We don't get as good presentation by MHC class 1. So we mostly get a humoral response. Okay, remember that humoral is with the B cells activating to make antibodies. So the antibodies are acting in the blood, the humors of the body. Generally, that's okay. Even a viral infection, right? The virus is going to be outside of the cell for a time, and those antibodies can neutralize those viruses outside of the cell. And again, generally, that's enough, even if you don't get good cytotoxic T cell activation. Okay, so here's just some examples. Influenza, we actually have an attenuated version of that as well that seems to work a little bit better for kids. But rabies, Salk polio, pertussis. The killed vaccines are either considered whole cell or subunit. So they're whole cell if we take, say, the whole bacterial cell and kill it. They're subunit if we purify just a portion, maybe some protein either from the surface of the cell or inside of the cell that's particularly antigenic. And there is one real drawback with a whole cell vaccine. Potentially inflammation is a problem, and this is with gram-negative cells. Because what do you know is on the outside of gram-negative cells, that lipid A, otherwise known as endotoxin. Okay, now when the bacterial cell dies, the endotoxin kind of gets released a little bit, but apparently enough of it is still associated with the cell to often cause a little bit more of a reaction, an inflammatory reaction. So the pertussis vaccine used to be given, we get it in conjunction with this D here stands for diphtheria, T stands for tetanus, and the P was pertussis. Okay, that's the form of the pertussis vaccine. It used to be a whole cell vaccine that we used to give. You know, when you give babies a vaccine and it makes them really cranky for a couple days, and if they have a fever and, you know, swell up their arm, mothers are not likely to want to give that vaccine again. And, you know, that's one that you have to have like 4 different times. So we've switched now to a subunit vaccine for pertussis. We now use the acellular pertussis instead of the whole cell. So now the form of that vaccine that we get is diphtheria, tetanus, acellular pertussis. So if you've seen that DTaP or, you know, DTP, you might have wondered what the difference was there. All right, now we give an acellular pertussis. It's just, I'm not sure which protein that they take from that bacterial. Again, that gram-negative Bordetella pertussis is the name of that organism that causes pertussis. But again, it's just just part of a protein, causes less inflammatory reactions. Unfortunately, it doesn't work quite as well. So you have to give more boosters, and it doesn't last as long. But if more people will get it, then you know, they've made the argument that this seems better. So little story. One thing that I did right as a mother, I had my son vaccinated again, because when you're a baby, you get pertussis vaccine, and you're supposed to have a booster when kids are about 12. Well, when kids are 12, you often don't remember to go and have these vaccinations done. But I remember to do that. And when my son, like 6 months after that, my son went to summer camp, and we got a letter a couple weeks after the camp that said, someone in your son's cabin came to camp with pertussis. He didn't know it, obviously, and somebody else in your son's cabin is ill with it. So if you're seeing these symptoms, you know, make sure you see the doctor. So these kids were both ones that had been vaccinated as babies. They just hadn't had their booster. It had worn off. And that was one time that I did something right. So I don't know, it sticks in my mind. Okay. All right, the last commonly vaccine that we've used for a long time, there are a couple of vaccines that we use that are considered toxoid vaccines. So there are a couple infections here, tetanus and diphtheria. Both of those infections, the ill effects are caused because the organism, the bacteria, produces a really nasty exotoxin. So it makes sense to try to stimulate antibodies against the toxin rather than the organism. Because we can use antibiotics to kill the organism, but then it's the effects of the toxin that last a long time and that are really potentially deadly. All right, so we inactivate the toxin. Again, sometimes they'll heat it, sometimes they'll hit it with some radiation to get a good antibody response. So that's pretty effective. These are both infections that we're probably not going to get to talking about later on. Of course, tetanus is a problem because that exotoxin, tetanus toxin, makes it so your muscles stay stimulated, like they can't relax. Okay, so that name lockjaw, you know, your jaw clenches up and it kind of radiates outward from there. Treatable when you catch it early on, but especially if you're traveling to some country where you're not sure you'd be able to get good medical care, they want to make sure that you're up to date on your tetanus toxin. That only lasts about 10 years. Okay, same thing with diphtheria. Don't get, long-term coverage with these vaccines. But diphtheria causes this pseudomembrane to form in the throat. It's especially dangerous for young children. And you would think, oh, just scrape that membrane off so they don't suffocate. But apparently that doesn't work. That you ulcerate your mucous membranes there. So again, potentially deadly because kids suffocate. So, all right, questions so far? All right, so we have a couple new kinds of vaccines that are kind of been provisionally accepted, or approved by the FDA, emergency use. Messenger RNA vaccines, the basic research that went into these goes back about 30 years. So we've been considering using messenger RNA as a drug, basically, and more recently as vaccines. So, and there are other messenger RNA vaccines that are in various stages of clinical trials that we've just kind of taken longer with. So these are really not brand new. I got this information from a really good review article that was written pre-pandemic, so 2018. And I can give you the link to that. It's quite long, but it touts messenger RNA vaccines as a really, these kind of vaccines have the potential to kind of bypass some of the problems that we have with other vaccines. We'll talk about that. And they also have the potential to be used not only for infectious disease, but for cancer. So that's really exciting. You can sequence maybe the tumor cells and then synthesize in the lab the next day a piece of messenger RNA that would make a protein that would target those tumor cells. So that's kind of an exciting application that, who knows, maybe we'll get to pretty soon. Okay, so obviously the Pfizer, the Moderna COVID-19 vaccines, Moderna was a company that name Modify RNA. So it was founded on this technology. And right after we sequenced the whole genome of the virus, the SARS-CoV-2 virus, almost right away within the next week, they had made a messenger RNA vaccine to it. So one of the really nice things about this type of vaccine, of course, and then had to test it and make sure it was going to be safe, is that you can very rapidly produce this kind of vaccine, and it's relatively inexpensive. I know you hear about the millions and millions of dollars that we're paying for this, but they're relatively inexpensive to produce. Many vaccines, like the influenza vaccine, we have to decide about 8 months ahead of time what strains we want to include in that, so we have enough time to make it and grow up the virus in eggs for the next flu season. All right, so if we had to do that for this disease, that would have been a huge problem. We can make the vaccine really quickly and really fairly inexpensively. And another advantage is it's really safe. So there's absolutely no way that you can get COVID from the vaccine. It's just a little piece of messenger RNA. And there's no way that any mutagenesis can take place. Okay, you know that you can't get messenger RNA doesn't change into DNA. All right, that's not the flow of information in our cells. So there's no way that our DNA can be changed from a piece of messenger RNA. Some disadvantages, though. For one thing, it took them a long time to figure out. There's problem. Of course, messenger RNA is quite a fragile molecule. We talked about when you look at the total amounts of RNA in a cell, messenger RNA is only like 4% of the total RNA of your cell because it just doesn't last very long. It's very fragile, very quickly broken down by enzymes within the cell and outside of the cell as well. So to try to get around that, that's why we have to store it at extremely cold temperatures. Okay, for the Pfizer one, it has to be stored at -70 degrees Celsius. right? So special freezers in most cases, had to be purchased in order to store that. Now it can be in the refrigerator for a short amount of time before it's administered. Why? But the Moderna one doesn't have to be kept that cold. And I tried to investigate that. Well, they're both messenger RNA vaccines. Why does one have to be really cold and one doesn't? Well, the formulations of those are secret because they want to be the only ones to make them, right? So we couldn't say for sure. Basically, the company tested it at different temperatures, and according to the company's data, that's what it needs to be kept at. So that's the best answer I can give to that question, why one has to be super cold and the other one doesn't. The other thing that we've done to try to keep the messenger RNA a little bit more stable is to use lipid nanoparticles to deliver it inside the cell. All right, so if we just tried to, just the messenger RNA by itself, again, that would just degrade really quickly. So we have this delivery system, lipid nanoparticles to try to get it into the cell. I don't really like the way I phrased this. I kind of did this in a hurry and posted it earlier than I should have. Worry about adverse reactions. Let's cross out adverse reactions and say side effects. Like I said, it is a very safe vaccine, but we do have some inflammation that's common due to innate immune reactions. Okay, so in other words, redness, fever, a little swelling at the site of infection, like those are all fairly common things with both of these vaccines. What they have done to try to cut down on that innate immune reaction is to, they substitute some of those nucleotide bases with a modified base. Okay, so they use modified nucleosides. And what that's done is try to, like, our toll-like receptors, say on our dendritic cells, recognize this messenger RNA as being foreign. Okay, so remember when the toll, this is basically a PAMP, a pathogen-associated molecular pattern, because it's viral. Right, so remember when those toll-like receptors recognize PAMPs, that trips like lots of aspects of our immune system, cytokine production. All right, so when we use some modified nucleosides, our toll-like receptors don't recognize that as readily and triggers less of an immune response. All right, so hopefully we'll get better at that, and eventually you won't have to worry about being sick for a day after you get a COVID vaccine, or hopefully, again, we'll get better at that. Okay. The other kind of vaccine, jump in anytime if you have a question or comment. The other vaccine that has got emergency approval is the Ch.17 vaccine. And again, it is a DNA vaccine. It's more oftenly called a viral vector vaccine. Okay, so what that means is it uses an adenovirus. And we mentioned the adenovirus back when we were talking about just viruses in general. It's an icosahedral virus, non-enveloped, it's got this nice protein capsid. So they used adenoviruses cause like common colds. So they've used an adenovirus that is not supposed to be able to replicate in our bodies. And then they put a piece of DNA in there that encodes for that spike protein, COVID. Okay, and I forgot to mention that before, because that messenger RNA, that piece of messenger RNA encodes for that spike protein, which is the most antigenic part of the SARS-CoV-2. All right, so we've got this viral vector, this adenovirus, and so you don't have to use these lipid nanoparticles. This protein coat of the virus protects that DNA for the spike protein and more efficiently gets it into cells. Again, the virus gets into cells, but it can't replicate. So this is a really nice article down here, just from the New York Times, just from a couple of weeks ago. It kind of walks through in nice, easy to understand pictures what's actually happening inside our bodies when you get a Johnson & Johnson vaccine. And basically, once it gets into our cells, so here's the DNA, it starts actually making, messenger RNA and then translating into that spike protein. So you get that spike protein actually presented on the outside of, whatever it is, a macrophage or a dendritic cell. And then you also get some presentation by MHC class 1 of kind of bits of that spike protein, being presented there. Okay, so you get, you're going to get both humoral activation, B cell activation, as well as T cell activation, because you get some presentation of that endogenous antigen by MHC class 1. Okay, and the DNA is a little bit more stable, so you don't have to store these vaccines at really cold temperatures. And the really nice thing for a fast rollout is that you only need 1 dose. Okay, still get some of the, inflammation and stuff that you're getting from the other vaccines, but definitely has some good things, advantages to it. All right, so a question that you've heard a lot is, or the comment that I've heard a lot of people say is that I don't want a vaccine, it's too quick, I don't trust it. All right, so we need to understand the basics of how vaccines are approved in this country. So I just took a screenshot of this page. This is from the CDC website of how vaccines are approved in this country. And before we even get to any kind of clinical trial, there's going to be kind of an exploratory phase, as well as testing in animals. Okay, so before that vaccine ever gets into humans, right, they've done some testing in different kinds of animals, look to see, side effects, safety, that sort of thing. But when it gets to the clinical stage, and I want to say when I was talking about this last semester, there was still like 180 some different vaccines in various stages of clinical approval. So we've narrowed it down quite a bit. In phase 1, you take a very small number of healthy volunteers, people that are willing to get the vaccine, as well as people that get a placebo. Right, so the manufacturer of the drug doesn't know who's actually getting it and who's getting a placebo. It's an independent agency that collects all that data. So a small number of healthy volunteers, and they look to see, okay, is the vaccine effective? Do we think it may be effective? Moreover, is it safe? And so does it have any adverse effects in people? If everything seems to be looking good, they move on to phase 2, which just means you're inoculating more people, so you get a larger sample size, you get a better idea of if it's effective or not, because of course these are not challenge trials. In a challenge trial, you vaccinate somebody and then you expose them to the infection. Okay, they've had to just kind of let that exposure happen naturally, so they needed lots of numbers to happen. They're going to mess around with the dose here a little bit and see what dose is most effective. Everything looks good. It moves on to phase 3. I know for the COVID vaccines, they were inoculating at least 30,000 people for each one of them, and then observing them for 2 months afterwards. Okay. Usually any adverse effect with any vaccine has always shown up within 2 months. Okay, there's no vaccine that has any kind of long-term effects. Of course, the autism thing in the 90s, that was a thing for a while, but that's been disproved over and over again. The guy made up the data that did it. So there's 3 phases. This Operation Warp Speed, the question is, did we rush through these phases? No, we did all 3 phases, but the time was sort of overlapped a little bit. So they started phase 1, it's looking good, let's go ahead and start phase 2, even before maybe everything was totally, all the data was looked at really closely. So they kind of overlapped. They still did all 3 phases, but they overlapped them in time a little bit. And they made sure that they had, again, that 2 months after phase 3. So, and then once they were approved by the FDA, then there's really a phase 4 that's ongoing, okay? And that's the VARS program. That stands for vaccine, vaccine adverse reaction reporting system. Okay, so again, this is a way, again, after approval, anytime anybody gets a vaccine, they get a sheet of paper. Here, these are some of the side effects that you might see. If you see this side effects, call this number or go to this website. Okay, there's a way to report if you have adverse reactions, and that way we can make sure that it's safe, kind of long term as we go. If you get a COVID vaccine, you should be able to scan in a code and they will email you or text you, like the week after or maybe longer. How do you feel today? Have you had these symptoms? So do that. Then they get more and more data about, who's getting symptoms and for which vaccine and all that. So that's an ongoing thing. Okay. So I would just finish with the points that, these 2 figures from your book, when you think about public health, keeping people healthy, there's really 3 arms of that in terms of infectious disease, right? Hygiene, good hygiene, having drugs to treat infections, and vaccines, right? Any infection that we don't have any drugs that work, like measles or polio, the vaccination has just been incredible in terms of lessening the number of people that get this. Okay, so when polio vaccine was introduced, polio has been eradicated in all but 3 countries. I think it's Pakistan, Afghanistan, and Nigeria. All right, so we're really close to totally getting rid of polio thanks to the vaccine. We're always probably going to have some measles because it's so transmissible. But, it used to be, if you ask your grandparents, did you have measles? And almost everybody had measles before the vaccine was out. So ask your grandparents the next time that you see them, numbers way, way, way down. And measles probably isn't as serious as polio, but there are definitely complications that go along with that can be really serious. So no question that, vaccines have done a lot in terms of helping us to stay healthy. All right, anybody have any questions, comments? Let me see, we got a question here. Fertility, okay. So is there anything about fertility that you have read regarding any of the COVID vaccines? Let me just address that. We have on our faculty, Dr. Renkema, some of you may have her for lab. She's an immunologist. And she has looked at that question concerning fertility, not in like Better Homes and Gardens, but in the primary research journals. And she understands better all the immunological aspects of all that. And she looked at that study that questioned whether COVID vaccines could harm fertility. And she said, basically, it was baloney. That there's no way that could happen when she went back and looked at that primary data. So I can't answer that very well, but from somebody that I trust about it, that understands those things, the fertility issue is just kind of not even a reasonable thing in her mind. So you could ask her if you know her more about that, and that's kind of the best that I can explain that. But thanks, that's a good question to bring up. Okay, let's see, let's move on then. Okay, so the end of the course, which we are approaching. We'll have after the exam on Wednesday, we only have 2 weeks left of class. We will take some time to kind of look at some pathogens. And it's kind of a nice way to end the class because we're going to get to kind of touch on things that we've talked on all semester. So it might kind of be reminded of things we talked about earlier, and that's always a good thing. So I will try to get a better plan up next week as to exactly which pathogens that we're going to cover. The last 2 times I've taught this class, I've done this part of the semester all online. So I have not as good a feel for how long it's going to take to get through things, but I'll kind of say, hey, these are my best guess. If we run out of time, we won't do this and that, whatever. But we're going to pick some bacterial pathogens and viral pathogens to kind of finish up the course here. All right, so I always like to talk about gram-positive bacteria, especially the Staphylococcus and the Streptococcus, not because most of them don't cause particularly serious infections, but they are so prevalent. And if you're in lab, you're somewhat familiar with those organisms as well. The Enterococcus, of course, we kind of consider a Streptococcus. It's very similar. We only recently split those two into different genuses. So I'm not really going to talk about them because they're just similar to the Streptococcus. Okay, so let's talk about staph a little bit. Let's see. Right. And again, it would be helpful if you're in lab here, but not totally necessary. So don't worry if you're not in lab. Of course, the name of the organism, coccus, that means they're round cells. And that prefix staphine means that they chain. Okay, so the morphology is round cells. The cell arrangement is that they chain together. All right, so I'll come back to that slide in a minute, but you may remember this slide that we covered earlier in the semester, just talking about some of that terminology. Okay, Staphylococcus, grape-like clusters. Sorry, I said that wrong. Streptococcus means they chain. Staphine means grape-like clusters. Right, just checking to see if you're awake. All right. Okay, so grape-like clusters. Where do you find them? A really good place to find staph is on your own skin and your mucous membranes. What other characteristics do they have? They are catalase positive. Okay, so catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. Balance that. All right, so if you're in lab, you should have done a catalase test here when you were doing your unknown. You put a little bit of the organism on the slide, you take it off some solid media, so you get a whole bunch of cells. You add a drop of hydrogen peroxide, and those bubbles are the formation of that oxygen. All right, so that's a test that we do to tell the difference between Staphylococcus and Streptococcus, because as we'll see, the Streptococcus aren't able to make that enzyme. Don't fall into the trap of saying, oh, it was catalase positive, so it must be a Staphylococcus. Remember, there are a whole slew of organisms that make catalase. Most aerobes will make catalase. Remember, it's a detoxification of this toxic byproduct, this toxic oxygen product. So lots of organisms make catalase. Our cells make catalase. Gram negatives, a lot of them make catalase. Staph do, strep don't. So it's a good way to tell them apart. Some of our staphs form capsules or slime layers. Okay, oftentimes it's really not thick enough to be considered a true capsule. They are, in terms of their oxygen requirements, they're facultative anaerobes. So very flexible in their oxygen requirement. They can do some kind of fermentation or anaerobic respiration as well as respire aerobically. And we do consider them a little bit more environmentally stable. Or environmentally a little bit more resistant to some environmental stresses. Now they can't form spores. They're not that tough. They don't have an acid-fast cell wall. They're not that tough. But in terms of being able to tolerate like temperature fluctuations or pH fluctuations or high salt, okay, our skin can be kind of a salty place. We got lots of staph on our skin. Then they're a little bit more environmentally stable, especially when you can compare them to the Streptococcus. Okay, and there's about 40 different species of Staphylococcus in this genera. We kind of forget that because in lab, we only talk about 3 of the more famous ones. But we are going to focus on one of those species, and that's Staphylococcus aureus, because it's the one that's really clinically the most interesting. In other words, it causes the most number of infections. So if you look at kind of where it occurs and how we pass it around to each other, it can be part of our normal microbiota. So again, kind of your nose, your mouth, your skin, not always and not necessarily in very big numbers. So it can be considered an opportunistic pathogen. They argue about that, scientists argue about that, whether to call it a true pathogen or opportunistic. If it's there, part of your normal microbiota, again, it can cause infection if the opportunity arises, but mostly usually you really need a really high infectious dose. It takes like 1 million cells in the right portal of entry to make you sick. All right, so commonly transmitted in hospitals, especially the strains that are antibiotic resistant, also by close contact in the community. So locker rooms, you know, daycares, health clubs, you know, anywhere where people are in close contact with each other. And there's a lot of skin infections so that it can cause. So anytime you're like sharing equipment that other people have used on their skin, then there's potential, if you have some kind of cut in your skin, to get some kind of staph infection, especially. All right, and here, this is what it looks like when we gram stain it, right? When we gram stain gram-positive organisms, they've got that little more peptidoglycan in their cell wall, and they're gonna stick with that primary stain, crystal violet. All right, so let's think about some of the things that make Staph aureus successful in causing an infection. Okay, we call these virulence factors. And there's a couple that are structural. I mentioned already that some of them will form some kind of a capsule or a slime layer. So here's looks like a TEM, a transmission electron micrograph, where you can kind of see the gray areas there is the capsule around the cells. What good does that do the organism? It helps them evade phagocytosis, especially by neutrophils. Apparently, macrophages are a little bit better easier, more able to engulf organisms with kind of a small slime layer. And again, if you're going to evade phagocytosis, then you're going to kind of tamp down on the cytokines that would recruit more neutrophils to the site, so inhibiting chemotaxis somewhat there as well. Okay, but that capsule does help it to colonize and form biofilms. You might hear of somebody that had some kind of surgical implant, like a new hip or something like that, and it got infected with some kind of biofilm. Could have been a Staph aureus biofilm. They're very difficult to get rid of. Usually have to take out the implant. And another structural virulence factor, protein A, basically, this is a protein that coats the outside of the Staph aureus cell. Okay, so down here in this picture, here's the Staph aureus, and it's got this protein A all over the outside of the cell. And it just reminds us that the outside of cells are pretty complicated places. There's a lot going on in the outside of the cell. So it's got this protein, and what protein A does, it binds to the FC region of antibodies. Okay, so remember the FC region is that stem. Okay, the fragment of antibody binding, that's the outside. That's the variable region. Okay, so if the Staph aureus can bind to that FC portion, the analogy I like is if you're going to catch a snake, how do you do it? You don't come at it, you don't face its fangs, you grab it by the back of its head so the fangs can't get you. So basically the protein A is grabbing the back of the antibody. So the antibody binding portion here, that's the portion that wants to adhere to the bacterial cell to cover up its adhesion proteins so it can't infect. All right, so basically you're neutralizing the antibodies so they're not going to do any good. They're not going to be successful in helping a macrophage or a neutrophil phagocytize this organism, because they would need to be bound this way to it, because then the phagocyte would get a hold of it like that. Okay, so this is again something that's helping the cell establish an infection. Okay, so let me just write that in to be more clear. Inhibits opsonization. Remember, opsonization means anything that's going to increase the chances that phagocytosis is going to take place. We'll say as well as complement activation by antibody. Remember that there's 3 different ways complement can be activated. One of them, classical pathway, involves an antibody bound to some antigen. All right. Lots of exoenzymes, the Staph aureus. And again, not every strain is going to be able to do all of these things. Certain strains might produce certain exoenzymes or exotoxins that we'll get to in a minute. Here's another one that you're familiar with if you're in lab, coagulase. So this is an exoenzyme that clots the blood and basically helps Staph aureus establish an infection. Okay, if it can kind of wall itself off from our macrophages and neutrophils that would be trying to engulf it, then it has more time to kind of reproduce and kind of establish itself. In lab, remember how we test for this? We inoculate rabbit plasma and incubate it. And if we get a little bit of a clot there, then that's a positive result. Okay. Remember I said there's like 40 or 50 different species of Staphylococcus. Staph aureus, there might be one other one that's kind of rare that makes coagulase, but it's mostly just Staph aureus. And that's probably the reason that it causes so many infections, because it's good at making coagulase. But it doesn't want to stay walled off forever. Eventually it wants to break out of that clot and infect, different parts of the body. So it's going to make staphylokinase that helps it to dissolve the blood clot. Another enzyme that we mentioned before, and we were talking about virulence factors in chapter 14, is hyaluronidase. This breaks down the matrix between cells. Okay, so it helps that bacteria just be more successful in invasion. And then lipase is an enzyme that hydrolyzes the bonds in lipids. So our skin can be kind of an oily place. We have those sebaceous glands, a skin infection that kind of invades those hair follicles. Organism's going to be necessary to produce lipase to kind of break down some of those fat molecules to be successful at establishing an infection. And we know some strains of Staph aureus can make beta-lactamase. Remember what that is? That is an enzyme that alters that beta-lactam ring of some antibiotics. Okay, so if a Staph aureus can secrete beta-lactamase, it can basically deactivate all those beta-lactam antibiotics, and it makes them able to be resistant to them. Okay, so MRSA, methicillin-resistant Staph aureus, that's a strain of Staph aureus that can make beta-lactamases. All right, I'll give your hand a rest here in terms of the exotoxins. I wrote everything on this slide. Remember, exotoxins only takes a little bit of them and they can work, they're very potent. So they work systemically around our whole body, and again, very small quantities. Staph aureus actually makes hemolysins. They are actually beta hemolytic. And we're going to talk about the different hemolysis types when we get to Streptococcus. But actually, the Staph aureus can totally lyse red blood cells. So you can see around the colonies here where there's blood in this auger here, and it's totally lysed those red blood cells. So you see kind of a yellow clearing around the colonies for an organism that makes these beta hemolysins. This is an effect that that seems to have more of an effect in vitro. So in the lab, we're not sure really what kind of effect that has in our bodies, but they probably can not only lyse red blood cells, but other kinds of cells as well. But again, we don't think that that has a huge effect in terms of the kind of symptoms that we see. Some strains make enterotoxins. Remember, not endotoxin. Endotoxins, lipid B, only gram negatives. Enterotoxin is a kind of toxin that causes nausea and diarrhea. It's heat stable. So what's the so what there? Say you have some food that's been contaminated by Staph aureus, and it's producing this enterotoxin. Well, if you heat up the food, you might kill the organism, but the toxin will still be active, right? So for that reason, if you eat some food that's been contaminated with enterotoxin, you can start feeling lousy pretty soon after you eat it, like within half an hour. You don't have to wait for that organism to grow up in your body. You can start feeling nasty right away. Some strains make an exfoliative toxin. Exfoliates, it does what it sounds like. It makes the skin separate and pull apart. So we'll look at an infection, pretty nasty, pretty painful, caused by a strain that makes exfoliative toxin. And then more rare is the toxic shock syndrome toxin. We'll look at the symptoms of that infection as well, but that toxin is a super antigen. Remember, we talked about the different kinds of antigen. A super antigen is one that is non-specifically activating T cells. So instead of just a couple of T cells being activated, like as many as 20% of the host T cells are activated. So it causes this massive cytokine production and can cause the patient to go into shock. So that means the blood vessels get too leaky, the blood pressure goes way down, not getting enough oxygen to the organs, potentially deadly. Right. All right, so let's, with the time we have left here, we can hit some of the Staph aureus infections. Again, some of the skin infections can start out, not seeming to be too dangerous. Some of them have the potential to become dangerous. Folliculitis is typically not a dangerous one. It's an infection of a hair follicle. So you see these kind of pimples, kind of pussy pimples around a hair follicle. You can see the pus there. So again, strains that produce the lipase that can kind of break down that oiliness of the skin are going to be more successful in causing a little infection there. More typically, you see this in areas of the skin that have been irritated, maybe by shaving, for example, or just by wearing tight clothes. Okay, but again, typically not a problem. Sometimes that Staph aureus can invade a little bit deeper, and that folliculitis can turn into a furuncle or a carbuncle. All right, so sometimes we also call these boils. So again, if virulence factors that help them invade a little bit deeper. Carbuncles are kind of a common area there is the back of the neck. And I don't know if it's because that's an area that just doesn't get washed as often. I don't know, but that's a common place to see them. Really, anything like this that you see, any kind of skin thing that you see that is not healing like you would expect it to. So a week has gone by, maybe 2, and it's not healing. That's something that should probably be checked out, because something like this has the potential maybe to go systemic. So you don't want to get that organism into your bloodstream. You don't want it invading other parts of your body. So sometimes a physician will kind of lance these, maybe kind of, drain them a little bit to try to speed the healing process, get some antibiotics on there. Okay, an infection that typically does not go systemic, that's not serious, caused by Staph aureus is impetigo. This is common among kids. It gets, just spread around pretty easy. Just a skin infection. It gets kind of pussy, scabs over, and then the scab falls off and it opens up again. So my kids had this and I thought, oh, they're just picking at themselves and that's why that's not healing up. No, it's because the Staph aureus, again, that coagulates. It's, our innate immune cells. We're not getting to it like they should. This takes a really long time to heal if you don't get antibiotics on it. And if it takes a long time to heal, then it's more likely to scar. All right. So again, just some prescription antibiotic cream will take care of that. Some kids have the tendency to get it over and over again. And then you got to just be diligent about cleaning that area with soap, more soap. But again, to keep it from scarring, you usually need to treat that with antibiotics. And of course, a much more serious infection is MRSA. And I just got this picture here from WebMD. I had to tell if something's MRSA. Can't necessarily tell by looking at it. Everybody, if they get, a cut or a bite that they don't remember, they think, oh, it must be a spider bite. I've read that spider bites are actually really not common at all, especially in Michigan. But MRSA would look kind of pussy, where a spider bite wouldn't. And of course, if you saw something like this, hopefully you would get that treated right away. Like all the hallmarks of inflammation there, that's probably painful. It's red, it's pussy, it's seeping, it's swelling a little bit. So something that definitely should be looked at. All right, so MRSA usually presents as skin infections, although again, you can have a MRSA infection as a consequence of surgery, some kind of surgery, and it's really serious. The incidence of these serious infections for some reason is going down. We don't really understand why, but again, they can definitely, have the potential to cause mortality. And I don't have good numbers of how many people have died from this. Sometimes it's not reported very accurately, and one source has wildly different numbers from other sources, but we do know for some reason it's not well understood that the incidence is going down a little bit. Okay, and anytime you get Staph aureus infection in the bloodstream, they can go to places that are, really serious. So bacteremia, another name for that is septicemia. Okay, that means in the bloodstream. And when Staph aureus is in the bloodstream, it can, for instance, get into your bone. So I didn't have this cited very good, but this was an Ethiopian girl that had a Staph aureus infection in her bone, and you can see how that is not the right shape. So she's not able to walk on that. It's very painful. You can treat that with IV antibiotics, but it usually takes a couple months to get better. Adults will more often have osteomyelitis in their vertebrae for some reason having to do with how the blood flows. But again, that's potentially a very serious infection. Staph aureus pneumonia is quite aggressive. causes about 50% mortality rate. So that's really serious. Endocarditis is an infection of the lining of the heart and the valves that can cause damage to the heart valves, really serious. And of course, meningitis is infection of the meninges, covers the spinal cord and the brain. So some potentially really serious infections caused by Staph aureus, usually not though, unless again, there's some kind of trauma or surgery or, it's gone septic. All right. We'll finish the staph infections a week from today and start strep. And I'll give you an idea of what the final is going to look like. Let me know if you have questions about the tests on Wednesday, and hopefully I'll see you then.